everyone. What you're going to view now is a tutorial on histology. Histology is rather a difficult subject and most students falter when looking at the slides. So what I'm going to do is walk you through some of the slides that have been linked to the lab, and I'm not going to go through all of them, but I'm going to go through some of them so you know what you're expected to look at. In this first part, what we're going to look at are the slides of some epithelia and connective tissue. Now we are going to look at simple, pseudostratified and stratified epithelia, and in connective tissue we're going to look at loose connective tissue, dense connective tissue, cartilage and bone. Let's begin by first looking at simple epithelium, and the simplest of epithelium is called simple squamous epithelium. Now, if you look at the images here, you will notice you have two images. The first is that of a blood vessel, and the second is that of the glomerulus of the kidney. Now, the first thing in any epithelium is the presence of the basement membrane, which is the area, it's a membrane on which the cells lie. Now, if you look at the arrows pointing, you can see that they are pointing to the basement membrane. And lying on the basement membrane are the nuclei of very, very flattened cells, and these are known as squamous cells. So you can only see the nuclei, they are flattened, and you do not see any cytoplasm. All epithelia have a free edge and an attached edge. And the free edge faces what is called the lumen, or the open surface. So here we have the arrow pointing to the lumen. And like I mentioned, we have an attached surface, which is the area where it's attached to the basement membrane, and the free surface is where it faces the lumen. So that is the simple squamous epithelium. Next, let's look at simple cuboidal epithelium. And again, you have the basement membrane. And how do you tell where does the basement membrane begin? You will notice that the basement membrane begins from an area where above the area the cells, or rather the nuclei of the cells, have a very orderly arrangement. So here too you can see the arrows pointing to the basement membrane, and lying on top of the basement membrane are the nuclei of the cuboidal cells. Surrounding the nuclei, in this you can see cytoplasm. This was something you did not see in the previous image, and this cytoplasm is present on either side of the nucleus, and very little cytoplasm actually is present above the surface of the nucleus, on top of the nucleus. Now again, here we are looking at the lumen, and you can see that the free edge of the cells faces the lumen. And if we look at some of the distinguishing features of cuboidal epithelium, the cells are as tall as they are broad, and there is very little cytoplasm present on the surface or above the nucleus. Now let's move on to simple columnar epithelium, and like the name suggests, they are columns. That's why it's called columnar. Once again, we have the basement membrane, and sitting on top of the basement membrane, you have the cells where you can see the nuclei very distinctly. The nuclei may be rod-shaped or they may be round. What is distinct about this kind of epithelium is that you see cytoplasm, you see a lot of cytoplasm, and you see cytoplasm above the nucleus, because remember, the cells are taller than they are broader. Again, you have the lumen where the cells are facing the lumen, that's the free surface of the cells. And as I mentioned, the cells are taller than they are broader, and hence you see a lot of cytoplasm above the nucleus. Now here what we are looking at, these are two examples of where you would find simple columnar epithelium. The first one, where you can see that the nuclei are rod-shaped, this is typically seen in the lining of the GI tract, like the stomach, the small intestine, large intestine. And in the lower image, where you see these round structures, these are cut sections of round structures, these are ducts. and these ducts are typically seen in glands, and usually the nuclei there are rounded, and in both cases anyway, you see a lot of cytoplasm above the nucleus. Let's now move on to an epithelium which is called pseudostratified, and for your purposes, we are only going to be looking at pseudostratified ciliated columnar epithelium. Once again, you see the basement membrane, and if you look above the basement membrane, you can see that there seems to be an orderly arrangement of nuclei, so you know that that is where the basement membrane is. And then you see multiple layers of nuclei. Now these multiple layers of nuclei, that's what they appear as. They are not truly multiple layers, because all cells touch the basement membrane. It just gives an appearance of many layers of nuclei. That's why we call it pseudostratified. Now we call it ciliated because you can see these thin hair-like processes present on the surface of those cells, and these are known as cilia, these hair-like processes, and they face the lumen, which you see over here. So these cilia, you should not make a mistake. It's very, very distinct. In the lower image, which is darker and a little difficult, you do see some structures in the lumen. That is because it's been taken from the nasal cavity. So this is just a little bit of phlegm which is present in the lumen. So that has nothing to do with the cells. Now in pseudostratified ciliated columnar epithelium, which is typically seen in the respiratory tract, the nasal mucosa and the trachea, you also see huge single-celled glands which are known as goblet cells, and that's what the arrow is pointing to, and these contain mucus and they help lubricate the structures. Now again, to go back to typical features of pseudostratified ciliated columnar epithelium, there's a false appearance of stratification. You see many layers of nuclei, but actually all the cells touch the basement membrane. Some cells are tall, some cells are short, and hence it looks as though the nuclei are at different levels, which they are, but it's not that they'll sit one on top of the other. So this is not true stratification. Let's move on to a truly stratified epithelium, which is called stratified cuboidal. You also have stratified columnar, but it is a very rare type of epithelium and it's usually seen in patches, so I'm not going to describe it over here. Now here you see true multiple layers of nuclei, because you see one layer and sitting on top of this is a second layer of cells. And in stratified cuboidal, usually you have two layers of nuclei, which you can see. Again, you can see the basement membrane in both the images. And the top image is one taken from a very large duct. The lower image is taken from a smaller duct, and it's also under higher power, the lower image. You can see the lumen again, and in the lower image you can see some material in the lumen because it's been taken from a duct. And this is called stratified cuboidal because the top layer of cells are cuboidal. The bottom layer of cells may be cuboidal or columnar, but the point what you can see over here is there are two layers of nuclei, which tells you that this is a stratified epithelium, and since the top layer of cells are cuboidal, we call it stratified cuboidal. And as I mentioned, the lower layer could be columnar or cuboidal, but that does not change what you would call the epithelium. If the top layer is cuboidal, you call it stratified cuboidal. Let's now move on to a different epithelium, which is known as transitional epithelium, and this is actually also called urothelium. Now this is again a stratified epithelium where you have true multiple layers of nuclei, but instead of having just two, you have many layers of nuclei, as you can see up here. You can see that these begin from an area which is the basement membrane, and you have the lumen, so you have an attached end and you have the free end. And if you look at the top layer of cells, you will see that the top layer of cells are very, very plump. That means they have a lot of cytoplasm surrounding the nuclei. And you will have to differentiate this kind of epithelium from stratified squamous, which we will see in the next slide, where you will see that the cells are flattened. So in transitional epithelium, the top layer, you must look at the top layer and see that the cells are extremely plump. That means they have a lot of cytoplasm surrounding the nucleus. In transitional epithelium, another fascinating feature is that on the top layer you have a thin membrane called the cuticle. And this helps because this not only protects the wall of the urinary tract, which is where the transitional epithelium is found, but it is impervious to urine. That means it does not allow water, which is present in urine, to pass through the epithelium and get inside the wall of the bladder. And that is called cuticle. So again, the distinguishing features of transitional epithelium: one, it's many layered, therefore stratified, and the superficial layer of cells are plump. Let's move on to stratified squamous epithelium, and we have two types. We have one which is called non-keratinized, which is seen in the top picture, and a lower one which is called keratinized. Now, both of them have a basement membrane. They have multiple layers of nuclei, and you can see there are numerous nuclei, and you can see as you're going up, especially in the top image, look how the cells are getting flatter and flatter. And if you look at the non-keratinized type of stratified squamous epithelium, you can see that the top layer has flattened cells. You can still see the nuclei. So that's a very, very distinguishing feature. Compare this to the urothelium or transitional epithelium, which you saw in the previous image, where you notice that the cells were very plump. Here you can see that the cells are very, very thin and flattened. Now in keratinized epithelium, what you see is on top of the cells, where you can see nuclei, on top of them, you see a thick layer which has no nuclei, and that thick layer is known as keratin, and that contains dead cells. So there are no nuclei. So you can see the difference between the two. In keratinized epithelium, on top of the layer of cells where you see the nuclei, you will see a layer where you do not have any nuclei, and that area is keratin, and it has what is known as a homogeneous color. It may be pink, purple, it depends on the stain. So again, to recapitulate, non-keratinized epithelium, you see flattened cells on the top, it's multi-layered, but the flattened cells on the top, you can see the nuclei. In keratinized, you have a thick layer. It may be thin when you have thin skin. You have, but that layer is homogeneous. You do not see any nuclei. Now let's move on to connective tissue, and first we're going to do loose connective tissue. And the commonest type of loose connective tissue is areolar tissue, and it's called loose connective tissue because it has a loose arrangement, and it's formed by lots of collagen fibers. You may see elastic fibers. You can see elastic fibers in the top image. In the lower image, it's a little difficult to find these elastic fibers. And then we have fibroblasts, which are the cells which lay down these fibers, the collagen and the elastic fibers, and you can see fibroblast nuclei. The lower image is a more magnified image, therefore you're looking at the fibroblasts. They look rather large. And you can also see what look like lymphocytes in the lower image. They have typical round nucleus. You hardly see the cytoplasm. Now, loose connective tissue forms packing material, and it's loose, and you see a lot of spaces in between. And this helps to form packing material, and those spaces are filled with fluid. The next type of loose connective tissue which we are looking at is adipose tissue or fat tissue. And the typical feature is you see large cells pushed together, and these cells look very empty, and the nucleus is pushed to the periphery. The reason the nucleus is pushed to the periphery is because the whole cell is composed of fat globules which make up most of the cell, and they look empty because when we process the tissue, the alcohol removes the fat. And people have described this as a chicken wire appearance, which is why I have this mentioned over here. So if you look at adipose tissue, it looks like a chicken wire or a wire mesh. Now from loose connective tissue, let's move to dense connective tissue. And again, dense connective tissues of two types. You have dense irregular, where you have collagen fibers bundled together densely, but the arrangement is in a haphazard fashion. Dense regular, as the name suggests, it's a more regular looking tissue where the collagen fibers lie in parallel bundles. And so if you look at the top image, you can see that the fibers are cut in different sections. You have some going longitudinally, transversely, obliquely, so it's in a haphazard fashion, tends to have more spaces in it, and in the dense regular connective tissue, it's more compactly arranged and very, very regular looking. Now you can see the collagen fibers. The arrow is pointing to the collagen fibers. And in the top image, you can also see the arrow pointing to the fibroblast nuclei. You have fibroblasts even in dense regular, it's just that we can't make them out clearly in the lower image, so I don't have any arrow pointing to them.